The overall goal of this procedure is to describe how to isolate pure and double-negative T cells from the mouse kidney. This is accomplished by first digesting the kidney in a collagenase solution. Next, the mononuclear cells are separated by density gradient, and the hematopoietic CD45 positive cells are collected by positive selection. Ultimately, the double-negative T cells can then be isolated by negative selection.
In 1998, our team found that immune cells, particularly T cells, contrary to current dogma, were mediating acute kidney injury. There were very few of those cells in the kidney and hard to study. So we developed a technique to better study those. We were surprised to find a population of cells, the double negative T cell without CD4 or CD8, that were in high population both in normal kidney and during injury. The detailed and improved technique to study these better and to look into the mechanisms will be described in this upcoming video.
We have been studying double negative T cells since 2001, and recently we joined hands with Dr. Rabb lab to modify the original method for isolation of these cells to get a pure population that can be used for different biological immune responses.
Although this method can provide an insight into the immune cells in the kidney, it can also be applied to other systems such as non-lymphatic tissues.
Before starting the surgery, first add 10 milliliters of collagenase D to a petri dish for tissue collection. Then spray the abdominal area of a euthanized